Virtually every life process depends on proteins. Proteins are made up of amino acids. Understanding the structure and chemical properties of amino acids is essential for understanding the function of proteins. In this lecture, we will focus on three parts. The first part, we will discuss the structure of amino acids. Second part, we will focus on titration. The third part, I'm going to present you clinical correlations of amino acids. Although over 300 different amino acids have been described in nature, only 20 of them are most commonly found in proteins. These amino acids share common features. Each contains a carboxyl group and an amino group connected by a central carbon or the alpha carbon. Amino acids differ one from another by the side chains or the R group. The nature of the side chains dictates the role of amino acids in the protein. Based on the side chains, amino acids are divided into three groups. In the first group, there are 9 amino acids. Based on the molecular weight, the smallest amino acid is glycine, and the largest amino acid is tryptophan. Let's take a closer look at these amino acids. Glycine is the most abundant amino acid in collagen. Glycine is also a neurotransmitter, specifically an inhibitory neurotransmitter. Release of this neurotransmitter is blocked by tetanus toxin, leading to spasm. Alanine is a small amino acid. It contains 3 carbons. After removal of this amino group, it is converted to pyruvate, a final product of glycolysis. 3 amino acids listed here are branched-chain amino acids. Defects in metabolism of these amino acids leads to maple syrup urine disease. This is a high-yield disease in USMLE. Methionine can be converted to S-adenosine methionine, or SAM. SAM is a methyl donor. Methyl group can be used for methylation of DNA, modification of mRNA, or synthesis of epinephrine and creatine. Methionine is also the first amino acid present in proteins synthesized by cells through translation. Proline typically disrupt alpha helix. This amino acid is also hydroxylated in collagen. Now, let's take a look at the last 2 amino acids in this group, phenylalanine and tryptophan. Defects in metabolism of phenylalanine leads to phenylketonuria, or PKU, another high-yield disease for USMLE. Tryptophan can be converted to serotonin, a neurotransmitter. Tryptophan can also be used to synthesize niacin, a component for vitamin B3. Defects in absorption of tryptophan leads to Hartnup disease. In the second group, there are 6 amino acids. Most of these amino acids contains either oxygen or nitrogen in their side chains, except cysteine. Most literatures put cysteine in this group, although cysteine contains sulfur in the side chains, and the sulfur is very weak in attracting electrons. Serine and threonine are typical sugar attachment sites in protein. Specifically, sugars are attached to the oxygen in the side chains. This process is called O-link glycosylation. Serine and threonine are also attachment sites for phosphate. Phosphate group is attached to the oxygen in the side chains, and this process is called phosphorylation. Sugar can be attached to tyrosine as well, but this process only occurs during synthesis of glycogen. Tyrosine can also be phosphorylated. Tyrosine is a precursor for dopamine, epinephrine, and melanin. In our body, tyrosine can be synthesized from phenylalanine. Asparagine has 4 carbons with an amide group, and this is the amide group. Asparagine is a glycosylation site, but different from serine and threonine. Sugar is attached to the nitrogen in the side chains, and this process is called N-link glycosylation. Glutamine has 5 carbons with an amide group. Glutamine is an important way to transport ammonium from most tissues to the liver. Cysteine contains thiol groups or sulfur hydryl group in the side chains. 2 cysteine can be joined to form a disulfide bond. Cysteine is a component of glutathione, a chemical used to remove hydrogen peroxide, a reactive oxygen species. The third group contains 5 amino acids. The first 2 amino acids contain carboxyl group in their side chains. In solution, these amino acids have 3 ionizable groups: carboxyl group, an amino group in the backbone, and another carboxyl group in the side chains. As we learned from organic chemistry, amino group function as a base. Nitrogen contains a lone pair of electrons. As a result, amino group can readily attract a proton and become positive charged. The charged amino group has a higher pK value. Carboxyl group, on the other hand, can easily shred a proton and become negative charged. Carboxyl groups have a low pK values. Compared with amino group that has higher pK value. The other 3 amino acids all contain nitrogen in their side chains. Specifically, histidine contains an imidazoles group, and lysine contains a primary amino group, and arginine contains guanidino group. Because of the nitrogen, these side chains function as a base. They can readily accept a proton and become positive charged, and they have a higher pK value than the carboxyl group. Aspartic acid has 4 carbons. It can be converted from asparagine. After removal of the amino group, through transamination, aspartic acid is converted to oxaloacetate, an intermediate of the citric acid cycle. Glutamic acid is very similar to aspartic acid, except it has one more carbon. It can be converted from glutamine. After removal of the amino group through transamination, glutamic acid is converted to alpha-ketoglutarate, an intermediate of the citric acid cycle. Histidine can be converted to histamine, a mediator of the allergic reaction. Lysine is an abundant amino acid in histone. It promotes interaction between histone proteins and DNA. Lysine in histone can be acetylated to control gene expression. Arginine, similar to lysine, is an abundant amino acid in histone. It promotes interaction between histone proteins and DNA. In addition, lysine is a precursor for urea. Here are the 3 letter codes for amino acids. Students are required to know these 3 letter codes.
Now, let's switch to part 2. We are going to analyze the charge of amino acids in solution.
First, I'm going to introduce you Henderson-Hasselbalch equation. Giving a weak acid, here represented by Ha, in solution, the weak acid can shred a proton, leaving conjugate base. At the same time, the conjugate base can accept the proton to form acid. At equilibrium, both reactions will proceed at equal rate. Therefore, no net change in concentration of acid or conjugate base. At this point, the proton concentration times concentration of conjugate base divided by acid concentration is a constant. We call this dissociation constant, or Ka. If we take the logarithm of the both sides of this equation and define pH equal to minus log proton concentration and pKa equals minus log Ka, we can easily obtain the following equation. And this equation is Henderson-Hasselbalch equation. The key parameters of this equation is pKa. How can we determine pKa? Okay, let's use acetic acid as an example to illustrate how we determine pKa. We put acetic acid in a test tube. We add sodium hydroxide stepwise, and we measure the pH before and after adding sodium hydroxide. At the end, the data is plotted in a chart. pH is plotted on the x-axis, and the amount of sodium hydroxide added is shown on the y-axis. And here is the curve. And we call this chart a titration curve. As we see from this curve, after adding sodium hydroxide, pH initially changed very rapidly. Then it changed slowly in the middle range. After passing the middle range, pH started to change rapidly again. From this chart, we get 2 pieces of information. First, a range in which pH changes slowly, the middle range, is called buffer region. In the middle of the buffer region, there's a turning point or inflection point, and that pH is pKa. This is how we determined pKa experimentally. So next we ask, what can we get from this Henderson-Hasselbalch equation? Let's look at acetic acid again. If we move the pKa to the left side, and the Henderson-Hasselbalch equation will look like this. For simplicity, we designate the acid form as a form 1 and conjugate base as form 2. When the pH is lower than pKa, this term is less than 0. Therefore, the form 1 concentration must greater than form 2. Right? That's the conclusion. Opposite, when pH is greater than pKa, and the form 2 concentration will be greater than form 1. When pH equals pKa, and the concentration of form 1 and form 2 are equal. The conclusions we have learned from acetic acid can be applied to any weak acid or bases. These are the major buffers in the human body. The bicarbonate carbon dioxide buffer is the most powerful extracellular buffer in the body because concentration of bicarbonate can be controlled by the kidney and the lung. The phosphate buffer plays an important role in buffering renal tubular and intracellular fluid. Proteins are important for buffering intracellular fluid. It also helps buffer extracellular fluid, although it works more slowly than the bicarbonate system. Henderson-Hasselbalch equation is also important for understanding physiology and pharmacology. For example, excess reabsorption of bicarbonate by the kidney can lead to an increase in bicarbonate concentration. pH will rise. This will cause metabolic alkalosis. On the other hand, pulmonary obstruction causes an increase in carbon dioxide concentration. pH will fall. This will cause respiratory acidosis. For the third example, I'm going to use a specific drug. Diethylcarbamazine, the drug of choice in the treatment of parasite infection. This drug contains nitrogen. In solution, this drug can accept a proton and become positive charged. If we designate charged form as form 1, the base as form 2, form 2 can be better reabsorbed in the renal tubular than form 1 because form 2 carries no charge. The charge present in form 1 make it difficult to cross cell membrane. When urine is acidic, form 1 dominates. Less drug is reabsorbed. When urine is alkaline, form 2 dominates. More drug will be reabsorbed. As a result, half-life for this drug is longer when the urine pH is higher. Now, let's look at titration of alanine. Alanine has two ionizable groups, amino group and carboxyl group, with two different pK values. When pH is low, both groups keep the proton. This is cation form of alanine, or form 1. When we raise the pH close to pKa 1, the carboxyl group starts to lose the proton. At this point, alanine has one negative charge and one positive charge, with 0 net charge. This is a Zwitterion form of alanine, or form 2. When we continue to raise the pH close to pKa2, the amino group starts to lose the proton. At this point, the net charge for alanine is -1. This is the anion form of alanine, or form 3. In the titration curve, there are 3 clearly defined regions. In region 1, pH below pKa1. Region 2, pH is between pKa1 and pKa2. Region 3 is pH above pKa2. Based on Henderson-Hasselbalch equation, we can easily determine which form of alanine dominates in each region. In region 1, pH below pKa1, form 1 dominates. Region 2, form 2 dominates. In region 3, pH above pKa2, form 3 dominates.
Now, let me ask you a question. In which region the net charge of the entire solution is close to 0?
The answer is region 2. In region 2, form 2 dominates. Form 2 carries 0 net charge. In this region, there's also a small amount of form 1 and form 3. When their concentrations are equal, their charges cancel out each other. The next question we ask is exactly at which pH the net charge of the entire solution is 0, and that pH is isoelectric point, or Pi. How do we calculate Pi? First, we look for Zwitter ion. Second, we look for 2 pKa's flanking the Zwitter ion. In the case of alanine, Zwitter ion is form 2. 2 pKa's flanking the Zwitter ions is pK1 and pK2. It turns out the Pi for alanine is the average of these 2 pKa's. Now, let's look at the amino acids that carry 3 ionizable groups. Here we use histidine as an example. Histidine has 4 forms. The net charge for these 4 forms range from +2 to -1. Please note, form 3 carries 0 net charge, so this is a Zwitter ion. Now, where's the Pi? Based on what we learned from alanine, first we look for Zwitter ion. And second, we look for 2 pKa's flanking the Zwitter ion. Since the form 3 is Zwitter ions, the 2 pKa's flanking the Zwitter ion are pKa2 and pKa3. The average of pKa2 and pKa3 gave rise to Pi for histidine. Using the same idea, are you ready to calculate the Pi for acidic amino acids? This will be your homework.
Amino acids have a wide range of clinical correlations. Here, I'm going to show you some correlations related to nutrition.
First, I need to introduce you 2 terms. One is amino acid pool. It refers to free amino acids available throughout our body. The amino acids pool is supplied by 3 sources. First one is from hydrolysis of dietary proteins. Second one is from degradation of body proteins. Third one are synthesized de novo from intermediates of metabolism. The ultimate source of amino acids is from dietary proteins. Second term, protein turnover. Most proteins in our body are constantly being synthesized and degraded. This is called protein turnover. We will discuss protein turnover in later chapters. Here I just want to highlight one point. Protein degradation can be accomplished in two ways. First one is through ATP-independent process mediated by lysosome. Second one is ATP-dependent mediated by proteasome. Among amino acids derived from hydrolysis of dietary proteins, 10 amino acids are essential. They are not synthesized in humans. These amino acids can be summarized in one mnemonic: PVT Tim Hall. It represents 10 essential amino acids. The other 10 amino acids are non-essential. Please note, the arginine is only essential in children. Nitrogen balance is an important concept. We need to know it for NBME. Under a normal condition in adults, the amount of nitrogen incorporated into the body equals the amount excreted. When the amount of nitrogen incorporated into the body is greater than the amount excreted, it is called positive nitrogen balance. This happens in several conditions. For example, growth, pregnancy, or recovery from injury or surgery. Conversely, when the amount of nitrogen incorporated into the body is less than the amount of excreted, it is called negative nitrogen balance. This happens in few other conditions. For example, protein malnutrition, we are going to see it in the next few slides, or dietary deficiency of even one essential amino acid, starvation, or uncontrolled diabetes. Kwashiorkor is an example of negative nitrogen balance. It is an acute form of protein malnutrition. Symptoms include hepatomegaly, as shown in this picture, edema, and ulcerative dermatosis, as represented by this picture. Patients have sufficient food, but they lack protein consumption. We should be able to distinguish kwashiorkor from few other related disorders. Two most encountered ones are anorexia nervosa. It is an emotional disorder. Patients have obsessive desire to lose weight by refusing to eat. Marasmus is an undernourishment. Patients don't eat enough food, typically because the food is not available.